Gone are the days where farmers have to solely rely on nature's mercy to determine the outcome of their labor. Today, technological advancements and scientific breakthroughs are revolutionizing agriculture, ushering in reform that not only promote yield but also a sustainable planet. Welcome to Gene to Grain, where science meets sustainability, a podcast where we delve into the transformative role of science in shaping future of agriculture and global food system. Join us as we explore our innovative challenges and a path towards more resilient and sustainable agricultural landscape. Good morning and Jai Hind. I am your host Samriddhi Mishra of BSc 3rd SEM, and today we are diving into the fascinating world of genome editing in agriculture. I cordially welcome you all to this episode of Samvad. We are privileged and delighted to have with us today Dr. Achuit Singh. He is a principal scientist at ICAR, Indian Institute of Vegetable Research. He is a biotechnologist with a vast experience and expertise in genome editing, transgenic crops, and promote a sustainable agricultural practices. Sir, it's an honor to have you with us today to share an insight on how science in lab can transform the life in the field and eventually to our dinner plates. Welcome to Gene to Grain, sir.
Thank you very much for calling me and to discuss regarding that genome editing. It's my pleasure to be with you. Yeah, thank you very much.
So, let's start with the questions. Sir, can you briefly tell us about your background and how you became interested in genome editing and agricultural science?
Sure. Thank you for this. Basically, I'm a plant biotechnologist and microbiologist. Working with the viruses started my work in research purpose. So I introduced by my teacher to look for the thing, and even the scenario regarding that vegetable crop, where the viruses is causing havoc in that case of causing disease in vegetables. So to understand these viruses and host pathogen interaction is completely molecular biology and plant work kind of thing. So we came to know the interaction, how the thing work in that, how the host defense mechanism work against this virus, and how the viruses suppresses the or silence the host defense mechanism. So while understanding the thing, it's really challenging part to make the virus control in that case. So these ideas and this work, research leads to understand the thing, followed by that technology in 2012, they come with that genome editing, particularly CRISPR-Cas I'm talking. Genome editing in case of CRISPR-Cas in 2012. That answer that many questions and fast kind of thing. It means to look for the resistance in the particular plant, vegetables, how to come and develop resistance, how to knock down the susceptible lines. So that led me to more interested to work in this area to updated technology for the genome editing. Why the genome editing CRISPR-Cas technology is used in a very prescribed way, and it's going to develop what we required at present, not only for us, even for the livelihood of the farmers also.
Yes, sir. Okay. So, what does your typical day as a scientist working in the field looks like?
Yeah, is when I start with the lab over there to start our research work, laboratories where we as the thing identifying the looking for the gene which we are working, but the plant for the development, and not only the laboratory work, we have to sit with the updated technology. We have to learn the updated technology while going through the literatures and the publication in that area, and as well as to look for the students to whom with working together in that part, and to discuss what we further look for in the other progress in that case. And as a laboratory scientist, it's not necessary to look around the field. I look maybe into concentrate into lab only. We look for the farmer's field. We go to our field also to look for the further answer in the natural condition. From there we think. So I try to visit. I always go to visit to the vegetable farm fields also, and sit with our colleagues and seniors for the future project goals. So like that, we have a day for the things in a day for all.
And so, how would you explain genome editing to someone with no scientific background?
Yeah, it's very, if I say, like very no scientific background, I say that most people are familiar with nowadays, students, even the teacher of the computer system.
Yes, sir.
It's a program where we want, like in computer, what the program is given over there. Suppose a program is been affected, and I just put it a Microsoft kind of office is over there. Like suppose you type on that thing, errors is happened.
Yes, sir.
So what are you going to do? You're going to do either delete or rectify this error. So genome editing is almost like that only. What we used to do, whatever the error is there in host plant, with the use of this CRISPR-Cas,
Yes, sir.
CRISPR-Cas, say a program or things, we modify the inside of program of the plant and remove. Once it is been done, we save it and move on. So like that, we just simply whatever the error is happening due to that plant is susceptible, the vegetable is susceptible to certain disease or certain abiotic stresses, we remove that part or we add that part to make the thing strong. Like same way the error in case of typing, you have to do the typing on the Microsoft Office, you find the error, mistake, just remove and delete and insert like that.
Sure, sir. Yes, sir. And so, how from restriction endonucleases to siRNA transfection to CRISPR-Cas9, how will you interpret this journey?
Yeah, that is really important. When we talk about restriction endonucleases, it's a basics in base is there. Because of that only we can move up to the CRISPR-Cas. So that very first is the recombinant DNA technology. When we restriction enzyme endonucleases, we used to play with that. We do look to modify. We used to insert. We do the cloning. Everything we used to for that to understand the things, and obviously the siRNA, what we call silencing technology. So with the help of this particular, we develop our vectors and the construct. We look for the RNA technology to silence the thing. When we silence the part, silence in sense, we look for the gene characterization feature, how this particular gene work. Like if I give the example, if I say this particular plant having the drought tolerance, and we want to this particular gene transfer into a variety which is susceptible. So how we can say this particular plant having the drought tolerance, unless we won't have a detailed study. We go for the detailed study with this gene and we silence that, mutate that with the RNA and silencing gene. We silence that particular the drought tolerance, identify after characterization, we silence this particular gene. Once it has been silenced, we can say that thing is responsible for to govern the system and the pathway, remaining pathway, other pathway. So this is the silencing part. And now the CRISPR, as I always say that CRISPR means a guided RNA. As I said earlier, means where the thing is required, this guided RNA is going to work as a GPS over there, and that remove that particular unwanted or insert wanted base pair or 1 to 2 or 3 or maybe particular there the gene kind of thing. So once having that restriction study kind of thing, endonuclease followed by your this silencing, now we look for the conclusion means we need a product. So in that case product, this genome editing technology, this is best at present right now. This gives the complete answer to final the product kind of thing in our hand in a fast and prescribed manner, and for the livelihood of the farmers.
Yes, sir. So, what are some of the most promising applications of genome editing in agriculture?
Yeah, genome editing is right now playing very important role in case of not only in case of only crop science, even in case of vegetables. The first edited plants come with the tomato. Those Japan has been released that part. Even in India, recently in May 2025, our Honorable Agriculture Minister has been released 2 variety of the rice. One is Kamla, and then that is from the developed by our IRI people and over there. So ICR has developed this variety to variety of drought. This is a tolerance to the drought and as well as salt. This not only give the drought tolerance against drought and salt, it's also giving the increase the production of the rice. So this technology is not only for that drought and salt tolerance, also it's giving the resistance against the viruses, bacteria, multiple disease resistance things are there over the thing. So definitely coming future we have numbers of lines of the crop which having that using this genomic technology will solve the many questions and answer with help of genome editing.
Yes, sir. So, what ethical or safety concerns are commonly raised about genome editing, and how do you address them?
Yeah, there has been regulatories for there. Government has given the very good policy. In March 2020, they have put it this genome editing under SDN1, SDN2, SDN3. So clearly they make a good policy under this part, and this is very clear and crystal clear how we going to work with the genome editing work, like SDN1 and SDN2, where we use the host modification genome, where we modify one base pair to many base pair with the using the CRISPR-Cas technology. So policies and obviously the scientific work together. It's definitely work. They works together, and obviously the original farmers who's been give the feedback regarding the thing. So together, all these three play. The hurdles obviously will be there. They ask the questions. Questions always been good for the development of the thing, like where the how the issues, how the edited plant going to work if there is foreign particles is present over there. So for that purpose, there is technology to confirm the thing that foreign particles is present or not. We are using a CRISPR-Cas which is been introduced from outside, but it's simply modified a base over there. Once its job is over, in next generation, it's going to be or next generation is going to be free from the plant. So that we have to prove.
Yes.
So obviously the as a workers and the policies maker, we have to clear the doubts on the basis, the doubts regarding that present of any foreign gene, any present of the repercussion of this modification. So everything is there in place as per the technology as well as the rule and regulation. We work accordingly.
Yes, sir. So, how can science and technology contribute to sustainable agriculture and food security?
Yeah, definitely. Science and technology is for the sustainable food development. For that purpose, I will divide into three. As I said, that science we do as a scientist. We develop a technology for that purpose, for the benefit of the human beings, and for the definitely in case of agriculture, we always look for the farmers. And policy, obviously, the policy is going to play important role. Policy means people how to whatever the science and technology is developed. Ultimate goal is to reach to this the original person. Original in sense like farmers' livelihood. So like technology and this science, we apply and we explain to the stakeholders and to the farmers to how these particular things going to help you in the future. Like that only is going to help you out.
Yes, sir. So, what role does policy play in enabling or restricting innovations like genome edited crops?
Not I say that a policy is going to play the hard. Policy is always benefit for the human beings, for the environment. Like I say the example, eh, as I said earlier, like scientist, policy, and farmers. We say a scientific, farmer, and policy all means they are correlated to each other. When scientific work in this area, the policy makers always look for that how to how to utilize this particular knowledge in a safe way. So safe way in the thing for the and to make the policy according not only for that particular concern of the workers, even for the farmers also, and the farmers the important is the farmers. Why? Because they are the original worker. They're going to say that what things are required for the fields. So accordingly, scientists work, and for the scientists, the policy going to make it right. Best example is our in for the genome editing policy in India and even in the US. In India, as I said, in 2020, they have released for the genome editing policy how the things going to work. So in India, the is really the policy is benefiting for the biotechnology and the plant biotechnologies workers, in particular in case of genome editing. That's why we able to our biotechnologists able to release the genome edited rice.
The world is more concerned toward genome editing
Yeah,
these days.
particularly India and US are looking in the good way, but India we are getting it. Europeans are little bit having their strict policy which hurdling the case of genome editing, but they will come. They are working in this area. They will come out with this part
And
also.
Japan is the first country to develop a
Develop
transgenic
the edited lines.
red crop. Yes, sir. So, how important is collaboration between scientists, farmers, and policymakers in driving agricultural reforms?
Yeah, I answered that in previous question also. The same way, as I said, scientists is for to develop to work the science, to develop the requirement on the basis of requirement of the farmers. Suppose I have developed something, a crop which is no use of the anything of the farmer. What going to be this technology? So unless we have to original stakeholder, we have to understand the what the farmers and what the fields are required in this area. So accordingly, we have to work, and we are working in that part. When suppose I give the example, like in case of tomato, the farmers are suffering with the leaf curl viruses. So we have started our work as a editing part to give the resistance against the TLCV viruses like leaf curl disease, tomato leaf curl. Second, the most important, obviously, is important the policymakers. How to smooth release of these lines to the farmers. They make the criteria, they make the thing, and they accordingly sit with the each and all these peoples, and they discuss the thing and make the policy to smooth and safe release of anything, because environment is more concerned for the future also. So like that. So all these three together, unless these three together, people know, policymakers, farmers, and as well as scientists, if they work these three together, they definitely will achieve our goals.
Yes, sir. So, if you could change one thing about the way agriculture science is perceived or handled globally, what would it be?
Yes, really very nice questions. So if you go with that, agriculture is always comes secondary. When what is the first is always the medical and technology when we talk about.
Yes, sir.
But most of people are completely, I will say most, I will say, I mean, all of people are dependent on the agriculture only.
Yes, sir.
We won't have a food, we can't make it out anything. We need a food. Morning, if you have a fast morning, in evening, we need at least food.
Yes, sir.
So I would like to make it is the primary agriculture for this thing. That's why I want to change, because the thinking medical is fine. When you have a good food, a healthy food, nutrition food, we won't need this kind much
Medical help or
attention
something.
to the medical part and other things. So that's why I want to change the thing.
Sure, sir. So, where can listeners learn more about your work or on the field of genome editing in agriculture?
Yes, I used to, whatever the things I learn, I get finding from there. I put it in our research paper also. It is available on my our website also, as institution. Even the journal website, they can learn what I am doing over in my lab there. And also they can free to come our lab and interact with me, and they want to learn the thing, come to our institution. Our institution is a public institution where is training programs are going over there, and where I give the lecture also for the students under the programs. So things are there available with my publication, with my interacting over there by on my mail and everything. They can learn from me anytime.
Yes, sir. So, then what ethical or safety concerns are commonly raised about genome editing, and how do you address them?
Yeah, genome editing always they concern, as I said, they look for the any confirm that any foreign particles are not there present over in their developed lines.
Yes.
So for that, There is, as I said, there is policy and techniques, technology has been there. So to confirm that if it is, I address that there is no any foreign particle in case of edited plants. So once we demonstrate the result, once the result been examined by the experts, it's not like that. It's been very policy, senior experts, they confirm that everything is there, then it's there. So there is a policy, I said, there is a very clear cut policy on that part. So ethical issues, there comes part the somewhere in the transgenic kind of thing, but in editing issue only what I have came to feel that is the presence of foreign like Cas9, I say the enzyme which is playing very important role, and antibiotics with marker we use, that definitely in the next generation is been knocked down, not out from that host plant. So these are the thing we used to do. Is not a very at present any complicated. The policy has been very properly made restricted, so we didn't face any at present any case of the genome editing right now.
Yes, sir. And that brings us to the end of today's episode of Gene to Grain, where science meets sustainability. Sincere acknowledgement to Sir for sharing his valuable insights, to all our listeners for joining us. Stay informed, stay inspired, and continue exploring the science for shaping our future. Thank you and Jai Hind.